Hi, I am Dr. Graciela Cremaschi from the Institute of Biomedical Research in Argentina, and I will refer to the work related to the dual role, this yin-yang role of thyroid hormones in cancer. Tumor microenvironment is the environment around the tumor containing immune, stromal, and hormonal factors that can modulate tumor growth and dissemination. On the last 10 years, we have been asking if thyroid hormones as hormonal factors of tumor microenvironment can affect tumor growth and spreading. To this aim, we have studied thyroid hormones' direct action on T-cell lymphoma cells. Human T-cell lymphomas are a heterogeneous group of aggressive lymphoproliferative disorders that can origin in immature T cells or can be the clonal proliferation of postemic T cells that can home in skin, and we have cutaneous lymphomas, or they can be peripheral lymphomas that are very aggressive forms of the disease with poor prognosis. We first evaluate thyroid hormone receptors in T-cell lymphoma cells, and when we talk about receptors for thyroid hormones, we not only refer to those nuclear receptors that bind T3 and lead to gene transcription, but also to those that mediate non-canonical effects described to be in the RGD binding domain of alpha-beta-3 integrin by Dr. Davids and his group, and then bind T3 or T4 with different affinities depending on the cell type, thus activating different signaling pathways like PKC or MAP kinase, including the activation of transcription factors and thus leading to gene transcription. Both nuclear and membrane receptors for thyroid hormone are present in TCL lines of different types of immature and mature origin. Only TR-alpha-1 was expressed in tumor T cells, and a high expression of the membrane TH receptor, namely integrin alpha-b-beta-3, was found in tumor cells when compared to normal T cells present in circulation. Overexpression of these receptors may suggest an enhanced response of malignant T cells to thyroid hormone effects. We then assess if these receptors are functional. Both T3 and T4 added at physiological concentrations were able to increase the proliferation of T lymphoma cells. This effect was maximum when both hormones were added together in physiological concentrations that occurs in circulation. Moreover, when using T3 and T4 coupled to agarose and thus being cell impermeable, not only able to interact with the cell surface receptor, similar effects were observed. Also, we found similar findings in all the other immature and mature T-cell lymphoma cell lines. And also, we observed these effects in three-dimensional cultures. The participation of alpha-b-beta-3 integrin as the membrane receptors of thyroid hormones in TCL cells was determined using cells with integrin alpha-b-beta-3 downregulated by interfering RNA. In all these cells, TH were unable to stimulate proliferation. We can see this both in immature and in cutaneous and peripheral T-cell lymphoma cells. And similar findings were seen in three-dimensional cultures when pre-incubating cells with a selective inhibitor of integrin alpha-b-beta-3 activity, silengitide. We can see that silengitide inhibits thyroid hormone's proliferative action. To determine the mechanism by which thyroid hormone via alpha-b-beta-3 integrins induce the proliferation of TCL cells, we analyzed the transcriptional changes induced by agarose-coupled thyroid hormones by RNA sequencing. We identified several genes related to proliferation, survival, and angiogenesis upregulated in these cells, and we focused on VEGF as its expression and angiogenesis correlate with survival and prognosis in peripheral TCL patients. To determine whether this association was present in TCL patients, we analyzed transcript abundance of integrin alpha B, beta 3, and of VEGF-A and B genes in a panel of primary mature T-cell lymphoma samples. We observed a statistically positive correlation between integrin alpha B or beta 3 levels and those of VEGF-B or VEGF-A in all the T-cell subtypes, thus suggesting a functional association in primary lesions. We then evaluate Thyrodormant's effects on breast cancer cells, as breast cancer is the most frequent malignancy in women and the second most common cancer worldwide. Previous studies from Dr. Davies' group demonstrate that T4 and the cell-impermeable T4 agarose can stimulate the proliferation of human breast cancer cells by increasing PCNA levels, and it depends on a membrane receptor-mediated signaling pathway, as a MAP kinase inhibitor was able to block these effects. We have studied the direct actions of thyroid hormones in murine triple-negative cancer cells, namely 41, and find that both T3 and T4 added together, but only in supraphysiological concentrations, were able to increase cell proliferation by cell titer blue assay and by bromodeoxyuridine incorporation assay analyzed by flow cytometry. But what happened with fluctuations of circulating levels of thyroid hormones in vivo and cancer? Have thyroid status any impact on cancer development and spreading? Studies derived from case control and population-based studies indicate that both hyperthyroidism and hypothyroidism influence cancer development. However, results are controversial, and the final consequence of this influence related to the incidence or aggressiveness of the disease depends on the sample population and the tumor type. But in none of these studies, the possible role of TH in shaping the anti-tumor immune response was even suggested. Several evidences have shown that alterations in immunity are frequently observed during physiological or pathological fluctuations of thyroid hormones. Our laboratory and others have demonstrated that hypothyroidism leads to immunosuppression, while hyperthyroidism increases immune responses, especially by upregulating the function of T effector cells. Based on these results, thyroid hormones would probably affect the immunological factors in the tumor microenvironment, thus shaping the anti-tumor immune response. So we decided to evaluate the influence of thyroid status on tumor growth and development in syngeneic models of tumor-bearing mice to evaluate also the associated anti-tumor immune responses. To this aim, we work with experimental models of mice with different thyroid status, euthyroid, hyperthyroid treated with thyroxine, or hypothyroid mice treated with the antithyroid agent PTU, and then inoculated them with syngeneic TCL or breast cancer cells so we can evaluate the associated anti-tumor immune response. When evaluating tumor growth in T lymphoma or breast cancer tumor-bearing mice, hyperthyroidism led to the largest tumors with the highest tumor volume and weight. T lymphomas are very aggressive tumors, and so, for ethical reasons, experiments must end at 15 days. In this model, no differences were found between hypothyroid and control mice. However, at day 35 post-injection of breast cancer cells, hypothyroidism also led to a decrease in tumor size. We then analyzed tumor spreading. We do not find spontaneous metastasis in the T lymphoma model at 15 days, so we performed the experimental metastasis test by inoculating tumor cells intravenously and assessing their ability to extravasate and colonize a particular organ. For TCL-bearing mice, colonizing in liver and kidney, we found that despite having the biggest tumors, a decreased number of hypothyroid mice developed metastatic nodules in the liver. Moreover, a greater number of metastatic lesions was observed in hypothyroid animals, indicating that the hypothyroid status favors the dissemination capacity of T lymphoma cells. Similar findings were observed for breast cancer dissemination. At day 35, we have spontaneous metastasis in lungs, and the number of lung metastases was increased in hypothyroid mice at this time point. However, in the experimental metastasis test, we also found a decreased number in hypothyroid conditions. Could thyroid status regulation of anti-tumor immunity account for the discrepancy between tumor growth and dissemination? We then analyze what happened intratumorally with the lymphocyte infiltrate. It means with local immunity. Here I'm showing the results obtained in breast cancer tumors, but similar findings account for TCL tumors. Tumor-infiltrating lymphocytes were higher in hypothyroid conditions, with highest difference at 35 days when tumors of hypothyroid mice showed a significant decrease in tumor size. I'm not showing here, but we found this was accompanied of increased number of activated T cytotoxic lymphocytes in hypothyroid tumors. So we analyzed cytokine production in conditioned media from breast cancer explants. Hypothyroid tumors displayed increased levels of interferon gamma than hyperthyroid and control mice, thus indicating an active cytotoxic activity within the tumor that would explain the reduced tumor size in these conditions. The largest size of hyperthyroid tumors would account to the low number of tumor-infiltrating lymphocytes, but also could be related to the proliferative actions of thyroid hormones that we found in vitro. Surprisingly, we found high levels of CCL2 or MCP1 in conditioned media of hypothyroid tumors, while the contrary was observed in hyperthyroid ones. Chemokine CCL2 is mostly produced by tumor cells and is related to cancer metastasis and invasiveness, homing mesenchymal stem cells that are immunosuppressive to tumor microenvironment. This gives us a clue to understand the increased tumor spreading under hypothyroid conditions. Then we analyzed the immune content of the draining lymph nodes, the first site involved in tumor cell dissemination, and of spleens, as they reveal systemic changes in the status of circulating immune cells. In these samples, we evaluate the lymphocytes component, T cell subsets, B cells, cell activity and or activation markers and cytokines, and of the myeloid component, we evaluated myeloid-derived suppressor cells. In breast cancer, we also studied mesenchymal stem cells, as this stromal component can be attracted to the tumor site by chemokine CCL2. For simplification, I will use this picture to summarize our main finding. In hyperthyroid animals, we found a systemic increment in the number and activity of natural killer cells and of CD8 positive and activated CD8 positive cells. The importance of T cytotoxic cells in preventing tumor spread was demonstrated as an increased number of renal metastases was found in hyperthyroid mice depleted of CD8 positive cells. We also found a decreased number of spleen myeloid-derived suppressor cells. In hypothyroid conditions, we found a diminished number of T cytotoxic and activated CD8 cells, not only at systemic level, but also in draining lymph nodes, where we also found an increment in the immunosuppressor T regulatory cells. This data would explain the greater spreading of cancer cells in hypothyroid tumors that were also accompanied by an increment of splenic myeloid-derived suppressor cells. By the other hand, in breast cancer tumors, In hyperthyroid mice with lower levels of chemokine CCL2, we found a decreased migration of the stromal component, mesenchymal stem cells, that are pluripotential cells able to differentiate, for example, to tumor-associated fibroblasts, thus generating an immunosuppressive environment. Based on our results, we can say that thyroid hormones can exert an increment of tumor size and angiogenesis through the direct activation of its membrane receptor integrin alpha B beta 3. But they are also crucial to modulate a favorable anti-tumor microenvironment that impair metastatic dissemination. Considering that T lymphocytes display a low or undetectable levels of this integrin, we can assume that thyroid hormones' effects on immune component of the tumor microenvironment are mediated mainly by their nuclear receptors. In this way, the addition of a selective inhibitor of the integrin alpha B beta 3, silengitide, would impair non-canonical actions, leading to a decreased tumor growth without affecting anti-tumor immunity. As an example of the therapeutic potential of our results, silengitide treatment was evaluated in 2 models of T-cell lymphoma patient-derived xenografts into immunodeficient mice. Animals that received silengitide treatment showed significantly smaller tumors compared to vehicle-treated controls. So to take home, thyroid hormones are important regulators of cancer development and progression, directly regulating tumor cell biology and the anti-tumor immune response. These results provide a potential therapeutic target for cancer treatment, the membrane receptor for thyroid hormones, and highlight the importance of monitoring the thyroid axis in patients with cancer. This is all my team. These are my collaborators. Thank you very much.